Morning, one and all, and today we are going to discuss about good morning, good evening, good afternoon, whatever time is it in your country. So here we are going to discuss about uh, basically we are going to discuss about the oops uh, we are going to discuss about lysosomes. What are lysosomes? Lysosomes were first discovered by a scientist named as Charles Dader. I'm not sure about the spelling Charles Dader, and he was the first scientist who discovered the lysosomes. And um, lysosomes are basically spherical bodies. They are spherical single membrane bounded bodies which are synthesized by the Golgi body. Basically, its synthesis is start from the very starting takes place from the rough endoplasmic reticulum. Then it is then its enzymes are made inside the rough endoplasmic reticulum. After which these are sent towards the smooth endoplasmic reticulum, from which these are sent towards the Golgi body. And inside Golgi body, modification takes place, and final product is obtained, which is known as lysosome. But that lysosome is having uh, inactive condition, which means it is in inactive state, and in inactive state that is known as primary lysosomes. Primary lysosomes. And when does it become active? We are going to further discuss that as well. So first, uh, it Golgi body makes the primary lysosome, and after that, what is its function? Its function is simply digestion. In simple words, its function is digestion. It is found in animal cells, found in animal cells, and in plant cells, what is which cells complete the function of lysosome? Which cell organelles? Sorry, which cell organelles complete the function? Vacuole, as well as spherosome and allurome. Spherosome, allurome, and vacuole uh, do the function of lysosome in plant cells. Now, coming back to our part, that here function is such that it is involved in digestion. Now, that can include intracellular digestion as well as extracellular digestion as well as cellular digestion. Um, Better to discuss it after some time. First, we were previously heading forward the primary lysosome. Now, in order to make primary lysosome a secondary lysosome, when primary lysosome combines with a particle with either a foreign particle or a cellular particle, it forms a secondary lysosome. Forms a secondary lysosomes, and that particle can include endosome, endosome, that can be phagosome, or that can be autophagosome. So these are the particles which can combine with primary lysosome in order to form the secondary lysosome. Now, in this case, um, what are the endosomes, phagosomes, autophagosomes? So first, as we know that we are having, for suppose this is a diagram of cell, we are gonna just uh, uh, inside that we are having this lysosome. This lysosome is having digestive enzyme. It is having some protein particles. It is having enzyme substrate complex inside it as well. It is having, its lumen and there's a membrane. So here, whenever a lysosome, whenever a cell, for suppose we are having a cell in which a particle wants to come inside. There's a particle outside the cell, and the cell engulfs this particle through endocytosis. Then this particle inside the cell would be endosome, and when endosome will bind with lysosome, it will be digested. Then it will be digested by means of what? By means of hydrolytic enzymes. What kind of hydrolytic enzymes? Such as amylase, protease, lipase are hydrolytic enzymes present inside the lysosome, and these enzymes are produced by rough endoplasmic reticulum in actual, which are further modified in Golgi body. So here, if this particle is entered through endocytosis, it is endosome, and of course digestion occurs. And if a particle is entered inside the cell through phagocytosis, it is phagosome. Then it is phagosome. Basically, if we are generally describing its endosome, if it is specifically defined, then it is phagosome. And when they combine together, they form secondary lysosome, where digestion occurs and digestive particles are then released into the cytoplasm either or just sent outside the cell. And this type of process is basically known as what? This type of process is known as heterophagy. This type of process is known as heterophagy. This type of process is known as heterophagy, in which a foreign particle outside the cell is entered inside the cell by phagocytosis, and then foreign particle engulfed in the cell by phagocytosis, and then it is digested, entered in the cell through phagocytosis, and then it is digested through the process of heterophagy inside the cell. That particle which is entered inside, foreign particle entered inside the cell by phagocytosis is digested through heterophagy is known as heterophagy. So here this is first step. Secondly, we are having another term which is known as autophagy. What is autophagy? For suppose we are having a mitochondria inside the cell. There's mitochondria inside the cell. Oops, a bit irregular shape, but it is mitochondria. So this mitochondria, for suppose, is present in excessive amount. This is basically a real situation that whenever we do exercise, the number of mitochondria inside our muscle cells are increased. So in order to produce more amount of energy, but after exercise the number of mitochondria are again sent back to the constant amount. Why does that happen? That only happens because of the process of autophagy. Autophagy is basically the process in which the lysosome digests its own cellular particle or reserve particle within the cell. And of course, in this process, hydrolytic enzymes do not come outside the lysosome, and autolysis auto sorry, it's autophagy occurs. Autophagy occurs, and autophagy is divided into two types. Autophagy is divided into two types, which includes the auto macrophagy first and auto microphagy second. What is autophagy? Macrophagy involves the digestion of large cellular particles such as large cellular particles such as mitochondria, cellular organelles such as mitochondria, ribosomes, etc. Automacrophagy involves small particles such as carbohydrates, proteins, and lipids. Apart from which, here uh, automacrophagy is further divided into two types, which is known as mitophagy and we have ribophagy. Now, mitophagy and ribophagy are what? Mitophagy is simply the digestion of mitochondria, autophagy of mitochondria, and that is autophagy of ribosomes or degradation of ribosomes. I guess it sounds clear. After which we are having another process named as um, we are going to further discuss that in intracellular, cellular, and extracellular digestion. So inside the intracellular digestion, we are having the following components. Autophagy and heterophagy are involved in the intracellular digestion. When it comes to the cellular digestion is the condition when enzymes which are present inside the lysosomes are shrinked out. The first of all, what actually happens in cellular digestion is that inside cellular digestion, for suppose uh, this is a lysosome, and inside lysosome, this lysosome will be having its membrane bursted. As its membrane will be bursted, the enzymes will be leaked out of the lysosome, due to which as these will come out of the lysosome within the cytoplasm, these enzymes will start digesting its own cell, and this time it will not be digesting the particles of cell but its own cell, complete cell, due to which this process is known as autolysis, or we can also call it as the process of cellular digestion, or we can also call it as the process of apoptosis. As we have previously studied what apoptosis is a process by which the number by which the cell digests itself, that is programmed cell death, which is regulated by the gene under the gene regulation system, time management system, or the genes which turn on and off system. And here apoptosis occurs, and after apoptosis, apoptosis basically occurs during the metamorphic stage as well. Metamorphosis, first egg is converted to larvae, then pupil, and then into adult. In metamorphosis of frog, when larvae, we can also consider larvae. In larvae, the frog is having its stage as tadpole. Tadpole is basically having a tail, but in the adult part, when it becomes adult, when it becomes an adult frog, its tail is disappeared. Why is that so? That is because of the process of apoptosis. During apoptosis, it the cells of the tail are supposed to digest themselves throughout their developmental stage, due to which when it comes from the stage of larvae to the adult, the tail is completely vanished due to the action of lysosomes during the process of apoptosis, autolysis, or cellular digestion. And this is only the process, apoptosis is only possible if we have a lysosome. But alternatively, in plant cells, there are vacuoles, spherosomes, aleuromes, as well as in prokaryotes, there are basically certain hydrolytic enzymes, not in lysosomes, but there are hydrolytic enzymes in the extensions of cell wall, inside the extensions of plasma membrane. These are present in the extensions of plasma membrane, due to which uh, these digest the materials inside the bacterial cell. And these hydrolytic enzymes are present within the cytoplasm and made within the extension of plasma membrane. Apart from which autolysis, apoptosis occurs, and there's another fact that during embryonic stage, during the developmental stage, embryonic stage, there are no gaps between our fingers um, inside the embryo, because there's a growth of cells within the spaces between the fingers. But as the embryo starts to develop, the cells start to disappear due to the process of apoptosis, autolysis, or cellular digestion, due to which the lysosome acts on its own cell and destroys it in order to form gaps between the cells of our finger, to form gaps between the fingers. And that is how it works, by destroying the cell, due to which there are gaps between the fingers. And after that, autophagosome is the particle. Autophagosome is a particle when the lysosome, when primary lysosome, autophagosome is a particle in which primary lysosome is fused with primary lysosome is fused with its own cellular particle or reserved food particle inside the cell, which is supposed to be digested through autophagy. Then it becomes autophagosome. I hope that sounds clear. After which cellular digestion is done, intracellular digestion is done, and lastly we are having extracellular digestion. In extracellular digestion, it is a digestion which takes place outside the cell, and in this type of digestion, enzymes are made inside the cell, but the enzymes are released outside the cell in order to digest the respective enzymes. For suppose we are having a sperm cell, and here we are having ovum. Oops. So basically, ovum is having two walls. First wall is known as the, which is the inner one wall, is known as corona radiata, and the outer known as zona pellucida, if I'm not wrong with the spell. And the corona radiata is made up of carbohydrates, proteins, lipids, and hyaluronic acid. Apart from which, zona pellucida is made up of glycoprotein. So uh, these are two walls which the sperm needs to conquer as well as break in order to enter the ovum. Now, a sperm cell is having basically acrosome in zone. Acrosome is basically a type of chromosome inside the sperm cell's head, which causes to allow or regulate the formation of two enzymes known as acrasine and hyaluronidase. Hyaluronidase is basically uh, hyaluronidic hyaluronic acid. Hyaluronic acid is basically an acid which is hyaluronic acid is the acid which was first isolated from the urine. This is why it is known as urinic acid. Hyaluronic acid. Hyaluronic acid because it was first isolated from urine. And now a formation of acrosine and hyaluronidase occurs here. Now acrosine is an enzyme which acts on zona pellucida. Hyaluronidase is an enzyme which acts on corona radiata. When both of these enzymes are released from the sperm cell's head from the lysosomes, then these enzymes act on zona pellucida and corona radiata respectively, due to which both of these walls are broken, due to which the sperm cell is unable to enter inside the ovum for fusion. This is how this process works, and that is an example of extracellular digestion, in which the enzymes are released outside and the digestion of walls of the ovum takes place, and these the debris of these walls is then excreted out from the female's body through urine. I guess that's all, and hopefully we'll meet again in another video.